Slide one, yeah.
And then we'll just do it on full screen so you have.
Okay, so thanks for the invitation. I think it's a great place to, I see many synergies between what we do and some of the presentations we had. The motivation, so we work in the field of omics, which is a big data field. And as many big data fields, progresses nowadays are driven by machine learning and in particular by trained predictive models. So one of the applications in genomics is answering basic biological questions relating genotype to molecular processes, in particular in gene expression. But you have also pervasive applications of machine learning and trained predictive models in variant calling, in designing experiments, etc. So there's a need to share those models. The field of omics has been extremely good in sharing data. So these fair data principles, I think, as a field we can be proud of, have been implementing them very nicely. There are also very nice efforts in standardizing software through open projects such as Bioconductor and Bioconda. But when it comes to those trained predictive models, we are not really there yet. So basically, those trained predictive models are usually shared in code repositories of individual groups. Sometimes you find them on paper supplements or other maintained web page. So the starting point of our project was for us to try to apply these FAIR principles now to this very particular class of software, which are trained predictive models. And this is our proposition for the field of omics called Kipoi. So it's a Greek word which means garden. And it's a collaboration between my lab and the lab of Anshul Kundaji and Oliver Stegle, and hopefully more groups in the future. So I'm going to guide you through these overview figures, which go through, on the one hand, on the left part, the contributor point of view, then the repository point of view, and then the user point of view. And I'd like to also directly now thank the journal Nature Biotechnology, which very nicely let this paper open access. That was a nice contribution from them, I find. So from the contributor point of view, the initial file you need to submit, and you may have only to submit this, is a model description file called model YML. I'm going to guide you through this. So the primary things that Kipoi stores is as models, and we see models as parameterized functions. And unlike software, here the parameters are fitted and they're fixed. And this is the thing that we want to share. Those parameterized functions can be implemented, typically they're in Python. You can also, of course, use any Python machine learning framework, and we are covering with our API all of those machine learning framework. And so what you describe in your model, YML, is the type of model you provide. And then these parameters are also called weights in machine learning. You share them as file on repositories such as Zenodo, which gives you then a DOI for those files and let them immutable. For machine learning frameworks, you can also serialize the architecture and as well store them on Zenodo. The other innovation we had for these models in the field of omics is to request contributor to not only share the model, which is a mathematical function from numbers to numbers, but also share the data loaders, which do all the pre-processing. It's extremely important for people to then be able to apply those models. We are providing with a package called Kipoi standard pre-processing, which start from standard files. So you have here the bed files, which define genome intervals, a FASTA file, which contains the sequence, and some functions in this package will then give you this so-called one-hot encoding, which is a standard way of representing sequence in machine learning. But of course, if your model has more complicated way to pre-process data, you submit on Kipoi the data loader. The goal is that your model will start from a standard file, and it's then very easy to apply it later on. For dependencies, we rely on Conda, and so your YML file has to describe the dependency via this Conda. You can also have pip in there. we check the predictions, and so on the contributor has to provide some test files, so here an NGF file, in which input and outputs are provided, and then we make sure that the predictions are reproducible. You also describe the schema, which means the dimensions of input and outputs of your model, as well as some description for users to know what they need to put in and what they will get out. And then there are also fields in this YML file that provide general information for the model so that one can quickly browse and find out what each model does. In particular, giving the authors link to the site, to references so that you can get cited. And also we've came up with a few tags that allow us to group the models by application area. So this is then automatically parsed and rendered on the Kipoi webpage so that users can have quickly an overview of what models do. Now, a bit more on the repository itself. So so Kipoi is essentially a GitHub repository in which each of the models is figured out as a subfolder. And then, if you have a very standard model which uses Kipoi for data loader, then you indeed only have to provide this YML file, and that's enough. If your data loaders are more complicated, you need to also provide them, and they would show up in your folder here. Altogether, we have already ported 23 model groups, which cover the major aspects of gene expression, so that we can apply it for those questions. And then when we go into the individual models, for example, on DNA binding, there are over 650 human transcription factors for which we have good models that can predict their binding to DNA. So if you go down to these single molecules, it's altogether over 2,000 models that are already available in Kipoi. You see this automated nightly test. So what we do is we test the models. This includes testing all the dependencies, so installing everything in a fresh conda environment. It includes checking all the pipeline from the input data via the data loader into the model, and as well the predictions match those provided in those test files. This testing is done upon any merge request, as well as every night on all model groups. Okay, so now the user point of view. Our API is implemented for Python, R, and command line, so that we cover a broad range of possible users, and we provide helpful functions to perform several tasks that I'm going to go now through together with you. So the first task is simply to predict, to use your model and apply it to predict. And that's very useful, as we heard, for benchmarking. So here I'm giving you an example on predicting the binding of transcription factor on DNA. So there are several classes of model out there, and these are 5 models that we've implemented in Kipoi. The first one you see here is a so-called position rate matrix scanning. It's a very simple model that looks at a very narrow region in the DNA. You have more complex models from the deep learning field, DeepBind and DeepC, as well as FactorNet, which takes on top of DNA also DNA accessibility. And we ported also a model based on a support vector machine. This is to say that you have the possibility to basically take any predictive models and share them in Kipoi. You're not linked to deep learning or so. Now, if you had to do this benchmark, you will spend quite a bit of time to figure out how each of these software needs the data to be preprocessed, make sure that this can be installed on your side. You will have to install the different software. When it comes to deep learning, machine learning framework, it can be a bit tedious. So the fact that we have done this once for all and ported them in Kipoi makes things then easier. So essentially, you just have these 3 lines of code to get any of these models. Here, this is a placeholder for one of these names here. Kipoi create and source activate Kipoi to have the model installed together with all the dependencies. And then prediction is done on very standard files you start from bed files and FASTA file. They are all parsed by the data loaders, sent to the machine learning model, and then the predictions are stored in HDF5 format. So this facilitates benchmarking, and in this case, we could show that all these deep learning models largely improve over classical models, of course. And okay, so there's really a need for the community, the end user community, to switch to this type of models, and we hope that with Kipoi we can facilitate the application. For those of you which use workflows, for example, SnakeMake, you will see that the fact that we have a single way to call each of these models facilitate the development of such scripts. Okay, another important application in genetics is to annotate variants for particular genomes These variants are typically stored in a file called a VCF file, which is a standard file format for variants. And because our models are all standardized, we could develop a unique plugin which works for all DNA sequence-based models of Kipoi. And so that facilitates the application of those models directly on VCF files, which are the files that are used in practice. So here I'm showing you what we do with in silico mutagenesis. You parse a VCF file. The reference sequence is extracted. The model is applied to the reference sequence, gives you a prediction, for example, whether you have a likelihood of transcription factor to bind at that place in the genome. And then we inject in silico the alternative allele. We run again the model, and we compare these two probabilities, do some transformation, for example, a subtraction, and store it as an annotation back into the VCF file. So that is done very conveniently with a single line of code for any type of models. So this facilitates the application of those models in pipelines. Okay, so we have collaborated with the company DNA Nexus, and they develop an applet that is based on Kipoi to do these annotations of VCF files. So this seems to be useful. Okay. One can also be interested more generally, not just at the effect of a single place, a single variant, but more globally within an area. What are the particular bases that contribute to a particular contribution? This is called feature importance score in deep learning, and so we have also a plugin that can generally, for all these DNA sequence-based model, apply techniques that compute feature importance scores, and we've implemented a few of those techniques. So here what you see, for example, is that at that location, that SNP is predicted to have a strong effect, but globally it is because it's centered in one of these GATA motifs, and it seems to be one of the most important places across the whole area. You can do this very easily for multiple models and see if all the models agree or not, which is more or less the case here at this location. Okay, another thing that is interesting now for people that like building new models is that you can leverage this repository to pull out models and then construct yours. And one of the most efficient way is using something called transfer learning. So transfer learning is the concept that if you have a model that has been trained for a particular task, it's going to be easier to tune it or adapt it to a similar task rather than starting from scratch learning. So if you know how to speak French, it's going to be easier to learn Spanish than if you start from scratch a new Roman language. So here what Anshul Kudaji and his team has done is develop a deep neural network which has been trained on 4021 cell types to predict accessibility of DNA. So it's a very large model that requires quite a heavy compute time to be fitted. And what we've shown is that once you start with this model and train a new model, but just changing the last parameters, but keep most of them frozen, you can get very quickly a pretty accurate model. So what is shown on this axis here is the time it takes you in terms of training epoch to get a good model if you start from scratch, if you relearn all the models, all the weights. And in this instance, it takes you over a day, whereas by transfer learning, it's in less than 4 hours, you have a model that is more efficient. And I show you this for this one cell line, but we've seen this very systematically for 10 cell lines. Applications on your own problems will definitely depend whether or not there are models in Kipoi that are close to your project, to your task of interest. But it's generally a good idea to start from some of those existing models rather than starting from scratch. And finally, having this repository of standard models, one can quite easily create ensemble models. So combine models to create new models. And this is a very well-used strategy to score variance. Most of the variance scoring methods out there are based on integration of different data and different predictions. Here we showcase this example. We showcase here this strategy for annotating variants that are near splice sites. So we are taking variants that are close to splice site and which, according to CleanVar, are either benign or pathogenic, and then train a model which is built on several Kipoi models that are related to splicing, and create quite quickly a better model. And so I show you the improvements of predictions. As you bring new model into this meta model, your ROC is, of course, increasing, reaching one of the very performant model at the end. And so this strategy, I think, is potentially for the community a better way to create new scores in which each group may focus on particular aspect of the biology, and then we can, as a community, openly create these these global scores. All right. So to conclude, I hope I could convince you that Kipoi aimed at have done some steps into implementing the FAIR principles for trained models in genomics. That this could be a good place to, as a community, create better models for doing this genotype to phenotype map. In the future, we hope to increase even more the number of models we have. One thing that we have in mind, and we've been talking to some challenge organizers, particularly the KAGI challenge, is to collaborate with challenge organizers by providing some naive models that are already Kipoi compliant so that users can directly, or challengers can provide their models directly in Kipoi. We are open for further groups to join us, to contribute models, or even simply to suggest which models they would like to have in Kipoi. And then we are looking already into how to extend the type of omics data modalities going beyond DNA sequence-based models, for example, count matrices, coverage tracks, or data from mass spectrometry. And another, I think, interesting direction is to integrate better with the bioinformatics community, and I think the talks that were before, and you, Salvador, goes very nicely hand in hand, and it would be great to make this solid. With this, I'd like to thank the people who worked on this, in particular Giga Offsets, who is here in the audience, and has been the lead developer, and I guess if you ask me 2 technical questions, I will point to him. Also Roman Kreuzhuber from the Stegle lab, the Kundaji lab. And if you're interested to join us, to join our monthly calls, feel free to send an email to any of us. I would be happy to open the door. Thank you for your attention, and I'm ready to take any question.
We have one question online from Spencer. How is the Kipoi scope defined? Your examples were from genomics, but would you accept other bioinformatics models, general machine learning models like GPT-2?
So I don't know GPT-2. It's genomics in the broad sense, so multi-omics is okay. So far we've been focusing on the DNA sequence-based models, and I think that is the scope. Yeah. Does it? I hope it answers, Spencer.
Yeah. Yeah. Go ahead.
I had a question about does the Kipoi repository require its users to also deposit their training data?
The contributors? No.
Okay.
So that is separate. The training data is something separate. That's especially important if you have things like if you train a model on GTEx and you have particular access rights. You need a dbGaP access to this. We didn't go into that.
And
therefore, also, it might be your question, it is not meant to support retraining.
right, that's where I was
Okay,
getting
this
at.
is your question. So the basic API doesn't do that. That's why also we can cover so many machine learning framework. You can still, if you're a Keras expert, take a Keras model and do your thing, of course. And that's possible. But this is not the goal and the main support of
Thank you.
KP.
And so we have one last question online. Do you do any benchmarking of different models of the same group? For example, variant calling? This is from Luis.
We don't do this yet, but I think that's a great extension. That would be indeed a good platform to allow for that. And the idea we have is to collaborate with the particular communities or challenge organizers to do that. We're interested in going into this, and we're happy to initiate collaborations along this line.
Thank you.
Okay. Right.